My name is Camille McCall. I am a postdoctoral research associate at Rice University working on the Houston Wastewater Surveillance Monitoring Program. For this module, I'll be discussing data management for quantification of microbial targets in wastewater. This is the third module in this series on quantification of microbial targets. To understand the steps and methods involved in microbial quantification, please see the first module in this series entitled Methods for RNA-DNA Quantification of Microbial Targets in Wastewater. Additionally, the second module in this series contains information on quality assurance and control measures for quantification of microbial targets in wastewater. The learning objectives for this module include to gain an understanding of what to consider when establishing a data management plan, to provide an overview of the role of a data analysis and management pipeline and the elements to incorporate when developing a pipeline for quantification data. And lastly, I will provide an overview of the data management pipeline used in our lab for the Houston Wastewater Surveillance Program. The data reported from wastewater surveillance programs are critical to the program's success. It can have serious implications if inaccurate data are reported. Therefore, in order to ensure the data has a high level of integrity, a data management plan should be established before sample collection and analysis begins. Things to consider when establishing a data management plan include the scale of the wastewater surveillance system, mainly the expected volume of the data collected. This consists of how many samples are collected and at what frequency sample collection takes place. The requirements of the stakeholders that will be using the data in downstream analysis, such as what files and formats are required. Goals of the end users. This includes the purpose for obtaining the data and how the data will be used, as well as any actions that will take place as a result of the data. Type of analytical data, for example, the Environmental Microbiology Minimum Information, or EMMI, reporting guidelines vary depending on whether the data are generated from qPCR or ddPCR assays. Data quality control measures should also be considered. For instance, how will the process controls be incorporated into your data pipeline, and how will they be used to flag things like contamination or inaccurate measurements? Finally, data security is an important aspect to any data management plan. When developing a data management pipeline, one should consider what measurements can be put in place to ensure the security and privacy of the data generated. A high level of data privacy may be required in cases where the data are sensitive and may be used to identify and stigmatize certain communities. A significant component of a data management plan is the data management pipeline, which consists of a series of steps designed to move data from its source to the desired destination, such as flat files, cloud storage, and end users. There are several characteristics of an efficient data management pipeline, including the ability to curate unstructured data, analyze the data, and provide sufficient storage. An example of this might be pre-existing and/or custom softwares that can gather and organize data from multiple sources, perform the appropriate calculations, and provide long-term storage for intermediate and end files. Additionally, generating and exporting data reports that consist of figures can help to draw insights from the data quicker and aid in decision-making. It is also important to consider how failed quality control measurements will be identified during data handling. In our lab, we incorporate our negative controls into our process limit of detection calculations, which helps us to easily and quickly identify contamination by visual inspection. And lastly, incorporating data collected through a laboratory information management system and integrating electronic lab documents are also important elements of a data management pipeline. The next part of this module will provide an overview of the data management pipeline used in the Stadler Lab. We'll discuss the inputs and outputs of the pipeline, as well as how we incorporate process controls and check for data quality. In ddPCR, targets are amplified using fluorescence-based probes. The template nucleic acid is divided into nanoliter-sized droplets, where each droplet functions as an individual PCR reaction. The droplet is then considered positive if the targeted sequence is amplified within that droplet. After amplification, the PCR plate is run on a droplet reader to obtain droplet data and perform further processing. What you see here is the Analysis tab of the Quantisoft software, where we analyze data from our droplet reader. This set of data is from wastewater samples run on a 96-well plate to target the SARS-CoV-2 N1 gene, represented by the green droplets, and the N2 gene, represented by the blue droplets. Before exporting the data, we go through a series of QC steps. We first check the accepted droplet counts for each sample, examine droplet clusters, and threshold the droplets into positive and negative clusters. Accepted droplet counts are checked to ensure each sample has at least 10,000 positive droplets. A droplet count of 10,000 is necessary for robust statistical calculations via the Quantisoft software according to the manufacturer. Shown here is a 2D view of the droplet clusters. As you can see, all the samples on the plate are highlighted and represented within the clusters displayed. While the green and blue droplets represent the N1 and N2 genes respectively, the orange droplets contain both targets, while the gray droplets do not contain the target. There should be clear separation between positive and negative droplet clusters as shown here. In the case of mostly scattered droplets, also known as rain, you may want to consider reprocessing the samples. Rain represents droplets with intermediate fluorescence and presents difficulty when classifying those droplets as either positive or negative. While there is no uniform cause, rain may be the product of sample impurities, DNA degradation, insufficient number of amplification cycles, or other issues. After inspecting the clusters, all samples are then manually thresholded by partitioning between positive and negative clusters. Quantisoft can then use the fraction of positive droplets to calculate the initial concentration of the target within a sample. Before exporting the data, we flag any samples or plate that fails QC and needs to be reprocessed. Samples are reprocessed if they display any of the following. 1, the accepted droplet count is below the recommended threshold. 2, there's evidence of contamination, i.e. negative controls with amplification. 3, unclear droplet separation, and or 4, significant differences in replicate concentrations. After thresholding and quality checking, droplet data is exported to .csv files and uploaded into our Google Drive Cloud workspace. The ddPCR data files contain raw concentration data generated from the droplet reader per each well and target. This information, along with various data sheets stored on the drive, such as the LIMS data, sample information data, ddPCR plate layout information, and other metadata, are read into an R analysis pipeline where the data is curated, analyzed to calculate the concentration, process limit of detection, and other data as appropriate for each sample simultaneously. Multiple benefits can be gained by using a fully or semi-automated analysis pipeline. First, it can keep the data analysis methods consistent, which helps to reduce errors and discrepancies in calculations between samples. It can process a high volume of data in a short amount of time and assist in keeping data records organized and accessible. The R pipeline exports several files, including a specially formatted file, which is sent to the Houston Health Department, reports containing time series plots, and the complete data sheet shown here. We perform a final QC check on the data before sending it to the health department. We check to make sure all samples and targets are present and concentrations are consistent between replicates. We flag any significantly high concentrations relative to other samples and take the appropriate measures to ensure the sample is representative of community levels. Finally, we check the limit of detection in our negative and positive process controls to identify systematic errors such as contamination. In summary, to ensure sample data has a high level of integrity, a data management plan should be established before sample collection begins. There are several things to consider when developing a data management plan, including the scale of the wastewater surveillance system, reporting requirements, goals of the end users, implementation of QC measurements, and data security. Data management pipelines are a series of steps designed to move the data from its source to the desired destination. Efficient data management pipelines are able to curate and analyze data, provide sufficient storage, flag failed quality control measurements, import and export data, incorporate information from laboratory systems, and electronic documentation. The data management pipeline used by our group contains a series of steps involving QC checks, integration into a cloud storage system/workspace, and analysis via a automated analysis pipeline. Using an automated analysis pipeline can reduce errors and discrepancies in sample calculations, process high volumes of data quickly, and assist in keeping data records organized and accessible. If you're interested in learning more about sampling, analyzing, and integrating wastewater data into public health interventions, check out our other training videos on our website. Support for this program was provided through a grant by the CDC. The Houston Wastewater Epidemiology team would like to thank you for viewing this training video.